So before I start, I wanted to give everyone a quick introduction on who I am. So thank you very much for joining me. I'm actually really excited to be presenting today. My name is Ruben Ajar. I'm the marketing director for the catalogs products for the GenScript corporation. And I've been with the company for the last 3 years. And I'm super excited because this was actually one of the very first projects that I worked on. And before that, though, I started my career very early on in actually a graduate research at University of Houston, where I did quite a bit of maxi-preps. And I got to experience not only how tedious they are, but also how time-consuming and problematic they can be. After that, I ended up working for the Human Genome Sequencing Center, where I got to see some of the challenges with laboratory workflows, and then I ended up joining the team that was automating the tedious manual workflows. So I got to experience a lot of the challenges of taking what is manual and automating it, and what that comes with. So basically any kind of compromises you have to make. And after that, I ended up working for a company that manufactures these large equipment to automate the process. So I got to experience the challenges that comes with that as well. So with GenScript, the nice thing about my experience has been to basically merge these various challenges, from the plasmid purification to the manual prep to the automation. So I hope that today what I present to you will be very informative, and I'll go ahead and dive in. So at the beginning of my slides, I'm going to be giving you kind of an introduction over what we have worked on, and then I'm going to be actually talking about the solution that we have developed, a couple workflow examples, and then some test data. And finally, I'll give you kind of a quick overview on some of the related products that we have. So those of you that are not familiar with GenScript, we actually have been around for 21 years. We were founded in New Jersey, Central New Jersey, in early 22. And we've come a very long way since then. The company was actually founded with just $5,000. And you can see it has grown significantly with quite a bit of different focus on different applications, mergers, acquisitions, things like that. And in late 2022, that's when we actually launched the solution that I'm going to be presenting today. So I don't think I need to present what the history of plasmid is, but I'll quickly touch base on why I have this slide. The reason why, of course, is that plasmids, of course, have been around for a very long time. And then the extraction methodologies that have enabled them to become a key aspect in really making the revolutionary work that they used with has really evolved throughout the last 20 years. So, of course, there's quite a bit of different levels of plasmid purification. We all have heard mini, midi, maxi, giga. But it's also important to mention that there's also the different uses of the plasmid. And with that, there's multiple levels of qualifications, right? If you're working with a research grade, you're probably working with a smaller amount. It's not going to be as large as a preclinical. You're also probably working with quite a bit of samples. Another thing is that you might not be as sensitive when it comes to quality. And of course, as you get to the industrial grade or the clinical grade, that's really when you get into the highly regulated or highly quality impacted methodologies. So this is kind of a quick overview of the different methodologies that are used for plasmids. Of course, I'm not going to go into that for the sake of time, but I do want to highlight here that when we have done a lot of our case studies or our test data, we've really tried to look at what the industry considers to be the gold standard, which of course is the column ion exchange setup, QIAGENs, the thermos, all those manual kits, because that's really what's been shown to give you the best results. However, with those solutions, as probably everyone in this room knows, it's extremely labor intensive, very tedious, and there's quite a bit of different centrifugation steps. So every time you add something to the tube or the column, you have to remove it, centrifuge it, back and forth. So it's a really painful process. And I remember when I did these back very early in my career, it would take me 4 to 6 hours to just do, a couple samples. And at the end of the day, the quality really varied. So it's definitely not only time and labor intensive, but it's also very variant. So with that said, really excited to be introducing today what we named the AmMag Quatro. And you'll see that there's basically 4 modules to the left and one little guy to the right. So that little guy to the right is the controlling module that controls every single setup that you're seeing to the left. So the idea here is that it's a very modular, scalable design. You can start with one purification module that can process up to 6 samples, or you can have this full 4 module setup. And all of these are controlled with that single controller. But if you choose to just start with one and you want to go ahead and add another one in the future, it's not a problem. Again, the idea here is that it's scalable, so it grows with you, it grows with your needs. Another thing to mention is that each module is completely independently controlled. So if you choose to run one module, let's say an hour, let's say at 10 o'clock, and you want to run the next one at 11, it's not a problem. Each one is independently controlled. And of course, you can run different protocols if you choose. One of the big things about automation is in a lot of cases, you end up having to optimize the solution quite a bit yourself, even though you could potentially have a manual solution that you've optimized. There's still additional optimization that you have to do when you switch to this different methodology. So what our team did has really tried to design this solution to make it pre-programmed or pre-verified for you. So basically you get the solution, you take it out of the box, you put it on your bench, and you're ready to go immediately. And you will get fairly good results. You can certainly optimize that. We do have the ability to do that. But again, the idea is that you can potentially take it out of the box, and out of the box, it's going to be ready to go. One of the other key features about the solution is the consumables. So the solution is comprehensive, and it will include the consumables, and I will go into that in a minute. Some of the main features as well, of course, is the high throughput. Whenever you're doing a maxi-prep, you're usually working with a few samples, but between, if you are, of course, in a higher throughput, you can potentially have that 4 module that I showed previously and run up to 24 samples at a time. So this is a pipetting instrument. So the precision of the pipetting is very important. And we were able to hit a CV of 3%, which is pretty precise. Another thing I want to mention here is that when you do load the instrument, the first step that you have to do is actually centrifuge your bacterial pellet, load it into the instrument, and then everything within that is basically completely automated. But the system is limited by that input that you have to load into the deck. And that usually, whenever you hear maxi-prep, you usually go by the volume, right? But that's not really how the AmMag Quatro's maxi-prep has been defined. Since we want to make sure that you're going to get the best optimal setup, what we recommend is you start between 1 to 1.5 bacterial wet pellet. So our range can be certainly less than that, but we certainly do not recommend that you exceed that 1.5 for multiple reasons, which I won't go into today because of time. But overall, you can imagine, if you stay within that range, you're going to end up with pretty good results. And then the other thing to mention here is that we took that tedious, very long process and we brought it down to about 2 to 2.5 hours. So you load up the instrument, you load up your consumable, you hit start, and within 2 to 2.5 hours, you're going to have transfection-ready plasmid. And when we say transfection-ready, we're talking about 2 main factors. The endotoxins, of course, which is less than 0.1 EU per microgram, and then the supercold ratio. And of course, that's really the form of the plasmid. Because, of course, I'm sure a lot of people here know that the form of the plasmid is going to be really dictating the quality of your transfection, because that's really the best form of the plasmid. As I mentioned before, the consumables is included, and it is all inclusive. So it's going to include everything that you need. And then the last feature I want to mention here is the user interface. Everything that you're going to need to run using the AmMag Quatro is pre-programmed, but if you choose to make your own protocol, it is something that you can do using the interface. Okay, so this is a quick overview of the consumable as well as how you would set up the instrument deck. You can see that basically A is the tips as well as the elution tube. B is the reagent cartridge that's going to include your magnetic beads, your buffers, as well as all the all the elution, the elution as well. It's also gonna include all the tubes as well as the waste. So everything that you're gonna use is basically included in the kit. And what's really nice about this kit is that it's actually significantly less than the industry average. So whether you're using a manual kit in the market or maybe you're using something that is a little bit more traditional, you're going to see a huge cost savings if you choose to switch to the AmMag Quatro. So I have this slide just to kind of give an overview of how the instrument works or how the process works. And I know it looks very complex, but it's actually, the idea here isn't to show you the complexity, it's to show you what's automated or how it works. So first, basically everything in blue, that's haloed in blue, it's completely automated by the AmMag Quatro. The first step that you have to do is to centrifuge your bacterial culture before you load it onto the instrument. So once you load it into the instrument, the platform is going to take you from the resuspension, the lysis, the neutralization, all the way from the washes, the binding of the plasmid, as well as the elution. So everything you're seeing in blue, again, is completely automated. The final step is going to include basically transferring the elution into a filtered column that is included as part of the kit. And the user will end up walking up to the instrument, capping that tube, and centrifuging it down. And that is certainly something that the user can skip. However, we do not recommend that you skip that because that's going to really get you to the lower level of endotoxins. That filter bed has that additional sterile filtration step, as well as removing any residual magnetic bead. Because usually when you do any process with a magnetic bead, there's going to be a little bit of floaters in the sample. So if there is anything left behind within the purification process, it's going to be completely removed by the filtration step. Now I'm going to give you a quick overview of our user interface. I think this guy is getting a little stuck. Okay, so I hope that earlier you guys remember that I had mentioned that every controller can control up to 4. So you can see in this particular slide here that I'm showing the interface, only one module is connected. The number 2, 3, and 4 are not actually plugged in, so you can see that they're grayed out. So using this home screen, this is where you can control up to 4 of them. I'm going to skip this one. One of my favorite things about the solution is that once you've set up your deck, when you walk up to, you're ready to get started, you're going to go ahead and scan the deck to confirm that you actually loaded it correctly. This is a huge pain point of automation, because if you don't load up your instrument properly and you go ahead and start it, and your sample is actually starting, whether it's lysis or pure part of the washes, it's too late, right? If you've made a mistake. So one of the really nice things is that instrument will scan the deck for you. It will give you a visual indication of whether or not you loaded it correctly. So you can see here, it's showing you the tips, the reagent cartridge with the barcodes, the reaction tube, the sample, as well as the waste. If you didn't load one of these, so you can load up to 6, you don't have to load all 6, it's going to say no in that particular position. Again, the idea is that it's giving you a visual indicator of what you have loaded and whether that's correct. So after you scan the deck, you'll go ahead and hit start, which is what you're seeing grayed out. And another thing that I find to be fantastic about this is that it will give you a countdown of how long it's going to take, dependent on the protocol that you loaded. So that means you can walk away and come back within that time frame, and that way the sample will be ready to go. You don't have to guess on when it's going to be complete. However, if you forget what time it started and you kind of need a reminder, you can again walk up to the instrument using the user interface. Look at the process, which is what you're seeing up here. You can see that in this particular example, the system has done the resuspension, the lysis, and the neutralization, and it's at the pre-clarification step. It has yet to do the supernatant transfer, all the washes, and then the elution. And it's giving you a little countdown, which is you're seeing here is 35 and 23 seconds. So it really is meant to be user-friendly. So now I'm going to be talking a little bit about the application data. And for the sake of time, I'm going to do it fairly quickly. So I hope I'm not going too quickly, but of course we can have a little questions about it towards the end. So the first example I have is to basically compare our solution, the AmMag Quatro, completely automated, and the manual kits that are available in the industry. As I mentioned before, this is what we considered when we were developing the solution to be the industry standard. So you can see after we did the purification on the Quatro using a vector with 3.1 and then another vector, sample number 2 with 3.4, we did the same thing with the manual kits. We are showing here that basically the ODs 260 to 80 to 60 to 30, as well as the endotoxin levels and the super cold ratio of the plasmid itself is fairly consistent with the manual kit. When you look at the concentration, one thing that the team did here when we set up this experiment or this data set is that they did a different elution. So that's where you see the concentration is a little bit different from the AmMag Quatro versus the manual. However, I do want to mention here, if you add the yield up, it's basically equivalent in performance. And then you can see the agar is gel to the right-hand side. Second one, I wanted to quickly give you a comparison of the different culture media. So, of course, plasmid prep is what yield you get is, of course, is how much you're putting in. So if you're working with a high copy plasmid versus a low copy, if you're working with an insert that's toxic, those are all going to be factors that are going to impede or impact how much plasmid you're going to get at the end. But another major factor is the media that you're using. So for example, if you're using a media like LB that's not very rich, it's not going to be as, of course, it's not going to have the conditions that a TB media would have, which is, of course, a lot richer, terrific broth than LB. So for this particular case study, what we did is 3 samples using LB and then 3 samples using TB. And you can see that the bacterial pellet weight that we ended up using, what we ended up starting with was 0.8 for the LB and then 1.2 for the TB. I've actually seen double that for TB. I've seen customers double what they have when using LB versus TB. However, the overall situation is that you do see that the concentration, the QC metrics like the OD, the endotoxins, as well as super cold, they're fairly consistent whether you're using LB or TB. However, of course, the concentration is significantly impacted. So for the LB, we got about 400 to 500 nanograms per microliter. However, the TB, we got closer to 800. So we almost doubled. So that's just a quick note on whenever you're trying to compare different things. Another case study I have is, this is actually was a customer sample. And of course, I got permission to use this. But you can see here that the customers chose a set of 6 samples, all of them different profiles. And what they started with 150 mLs of culture. And what they ended up having a pellet weight, it was between 1 to about 1.3, 1.4. And what we ended up doing is that we took these purified plasmids and we sent it back to them, and they performed a transfection for a protein expression, and they measured it side by side with the manual kit that they had purified with. So you can see to the graph on the right-hand side that for plasmid 1, 2, 3, they were basically equivalent or even better for the AmMag Quatro. And then 4, 5, or 6, again, a little bit less or equivalent to the manual. So with that said, the idea here is I wanted to show you kind of the purification and into the actual protein expression sample. So it gives you kind of an idea what to expect, which is equivalent, if not potentially better performance. Next case study I have is using the luciferase reporter gene. And what you can see here is we did a similar setup to before. We took the plasmids that we purified using a manual kit, and this was done actually by our services team. Our services team is really well known for high quality plasmid services. So this is another example of using an industry standard. And what you're seeing here, the little point, the blue one is the manual solution. The green and the purple is the AmMag Quatro, and then the brown one is the HEC 293 cells that did not get basically transfected. So what you're seeing here is compared to the manual or the kit, is that the performance was actually fairly higher to show using this case study. And then the last case study I have for you is using a PLNT vector. So what our team did is very similar to before. We did 2 sets of experiments where we purified using the AmMag Quatro and then another one using a control group, which was a manual. And I purposely I chose this particular example because if you look at the table on the top portion, you'll see that the concentration for the PLNT using the AmMag Quatro is actually about 300 nanograms per microliter. However, the control group was eluted in significantly less, 500 microliters. So it was 3 times the concentration. And then what we did is we took those 2 separate samples and we transfected them. And again, the idea here is I wanted to show a very low concentration for the AmMag Quatro versus a triple concentration for a manual prep. And I wanted to compare the titer. And you can see the bottom table is showing the control with sample 1 and 2, and then the AmMag Quatro with sample 3 and 4. And even though we transfected with basically threefold with the control group, we got almost equivalent results. So the average titer, which I have a circle of red around it, was about 53 to 49 for the control group, and then the AmMag Quatro was about 50 to 52. So pretty good results, especially when you're basically using 3 times the concentration. All right, so now I'm going to show you a quick video. Before we hit start on that, I do want to mention a couple things. The instrument has been developed to have the magnetic modules incorporated into the solution, as well as an automated mixing. The mixing is actually clockwise and counterclockwise. This is how we get really, really pure plasmid because of that mixing. It gets a really good vortex in the sample. And then the second thing I want to mention is also that the consumable kit is completely sealed. So that way, when you do load it into the instrument, you're not potentially having cross-contamination. It's completely sealed. You close the instrument and you hit start, and the system will actually puncture those foils that are sealing that the consumable kit. So that really maintains the sterility of your sample, as well as the making sure that you're not having cross-contamination between samples. So. Sorry about it being so quiet. There's supposed to be music, but that didn't come through. Okay. I can't, it doesn't look like I can control the slides anymore. Okay, so before I conclude my talk, or we start maybe taking a couple questions if there's any, I quickly wanted to show you our full protein workflow. We do have another system that's completely very user-friendly, also magnetic bead-based. And from what all the discussions I've had is that it's extremely user-friendly as well. And it's automating the protein or antibody purification. It's more semi-automated than the AmMag Quatro that I have shown you, but it's still very beneficial. We also have a full suite of electrophoresis solutions, including smaller devices as well as the precast gels and the consumables that go with it, including staining system, transferring, and an automated Western solution. And with that said, I wanted to give you a sneak peek into something that we're working on right now. This is what we're going to be launching hopefully at the end of this year or potentially the beginning of next year. And the idea of this system is that it's going to be a larger scale protein purification or antibody purification. And what you're seeing here, again, of course, it's going to be magnetic bead based. I think you know, Janscript has really developed or evolved their capabilities into becoming very competent with magnetic bead technology. So the vision here is that this is going to be a protein purification solution, and it's going to be configured in a way where you can probably see where I have buffer solution or station listed up there. You load the solutions from the top. The capacity is a lot larger than the AmMag assay that I just showed you. And that another feature is it's going to have a shaker incubator, as well as a reader that will enable you to basically read as you elute your sample. With that said, I just want to quickly invite you to the booth. I know that a lot of you probably have, I've heard from some people that I've spoke with today, they aren't aware that there's actually two entrances. So I put a little box in blue to the entrance on the right. So if you walk into that second entrance, you'll see us fairly quickly to your left-hand side. But if you end up coming in from the first entrance, you need to walk a little bit further to the right. And with that, thank you very much.
Questions?
Hello, thank you for your talk. I was wondering what the lowest solution volume would be for the, with those tips, and if it's just those tips or are there other tips that you can add?
So the tips are included in the kit. So it's not like you have to buy anything else, but it's just those tips. To answer your question, which is actually a really good point, thank you for bringing that up. With magnetic beads or any kind of elution, you really need to make sure you're saturating either the column that you're using or the magnetic bead itself. So it really is a ratio of how much magnetic beads you put in. The instrument itself can go all the way down to 500 microliters or even less, but it really, really is dictated by the magnetic beads that you use. If you end up going to a lower ratio than we recommend, you certainly can. But if you don't cover those beads, you're kind of sacrificing some of that plasmid that's on that magnetic bead, which could be okay for you, right? If you have enough plasmid on that magnetic bead and you just want a higher concentration.
Other questions?
All right, thank you. And by the way, I left my business cards, if anybody. And we do have an instrument at the booth if you want to check it out.
Okay. And also, thank you very much for your better explanation of the two parts of the exhibit hall downstairs. So think left, right. Thank you.